Hello everyone and welcome to today's session, and today we'll be discussing the chapter Biomolecules. Okay, so today we'll be discussing the NCERT section 9.1 and 9.2. Okay, and this is the part 1 of the lecture. Before starting the lecture, just want to give you one information that the images, text and other materials used in our presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. तो देखिए इस सेशन के ऑब्जेक्टिव्स क्या हैं? Okay, so objective of today's session is to understand the methods to detect the elements in the living tissue. So see, the living tissues as well as the non-living tissues, they are made up of these elements. So we will discuss how can we detect these elements in the living tissues. Next, we'll also learn about the qualitative test for various biomolecules. So see, biomolecules, okay, these are carbohydrate, proteins, lipids, DNA, okay, nucleic acid. So DNA is a nucleic acid, RNA is also a nucleic acid. So these are all biomolecules. So we will learn about the tests which are used to detect these biomolecules. We'll also list different type of biomolecules and their composition. Next, we will define primary and secondary metabolites. So there are two kinds of metabolites, primary and secondary. We will understand their functions as well. We'll also differentiate between primary and the secondary metabolites. So today, first I will give you a brief introduction of this chapter, biomolecules. Okay. Then we will discuss the topic how to analyze chemical composition, which is NCERT section 9.1. Then we'll also talk about primary and the secondary metabolites, which is NCERT section 9.2. Then we will summarize this entire lecture. We'll discuss some NCERT based questions. Okay, we'll discuss some keynotes and there'll be some interesting questions that will enhance your critical thinking ability. Okay, so let me first give you introduction of this chapter. Okay, so when you go outside, you can see there is a lot of biodiversity. You can see variety of life forms present on the earth. Okay, so there is wide diversity in the living organisms within the biosphere. Now, what is biosphere? So this term biosphere, what does this mean? So biosphere means all the ecosystem which are present on the earth. Okay, so how many type of ecosystems are present on the earth? So you have all the ecosystems are basically divided into aquatic and the terrestrial ecosystem. Then within aquatic you have many type of ecosystem, then you have many kinds of terrestrial ecosystem also. So all the ecosystems which are present on the earth and the living organisms which exist in these ecosystem, they constitute the biosphere. Okay, so they are saying there's a lot of biological diversity in the biosphere. There's a lot of species of insects, you can see there are a lot of species of plants, there are a lot of species of animals. Now, a common question arises here, whether all living organisms share the same chemical composition. Okay, so the question is that whether all living organism is made up of same elements, same molecules. Okay, so is it true or not? So we'll find out. So elemental analysis identifies elements and their mass content in the living tissues. So living tissues can be plants, okay, part of the plants, it can be animals, It can be microbes. Okay, all are living organisms. Okay, so let's see what all elements are present in the living, and we'll also talk about elements present in the non-living things also. So like elements like carbon, hydrogen and oxygen, they are found in the living tissues. So you can see living organisms, they are made up of carbon, hydrogen and oxygen. There are other elements also, but mainly they are made up of these elements. Okay. So these are the type of biomolecules you can see. Okay, you can see lipids here. Then you can see nucleic acid. Nucleic acid is both DNA and RNA. Then you have carbohydrates. Okay, and then proteins, which are made up of amino acids. So these are all the kind of biomolecules. These molecules, they are made up of carbon, hydrogen, oxygen. Okay. Now let's move on. Okay, non-living matter. Example of non-living matter, they have taken earth crust. It contains similar set of elements. So the elements present in the living tissues, these elements are also present in the earth crust. Now what is the difference then? So difference is in their composition. Like living tissue, they mainly consist of this carbon, hydrogen and oxygen. Okay, these elements are also present in the earth crust, but the concentration is different. And no absolute differences in elements between living tissues and earth crust. So what do you mean by this sentence? So this means that similar elements are present in non-living things and living things. Both are made up of similar elements. Now, living organisms have higher relative abundance of carbon and hydrogen compared to earth crust. So the living organisms, they are basically made up of carbon and hydrogen. So let's have a look at the table 9.1. We'll look at the table 9.1. So table 9.1, this is a comparison of elements present in non-living and the living matter. Okay. So here on the left hand side, you can see the elements are listed here. So elements are hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium and silicon. So all of these elements are present in non-living things, which is the earth crust, and living thing as well, which is the human body. Okay. You can see percentage is also given here. Percentage weight of these elements in earth crust and percentage weight of these elements in the human body. So let's talk about hydrogen. So in earth crust, it is 0.14, but in human body is 9.5. Okay. Then let's talk about carbon. Carbon is very less in earth crust, but it is very high in the human body. It is 18.5%. Then oxygen. In earth crust, it is 46.6%. In human body, it is 65%. Then nitrogen, it is very little in earth crust, but in human body, nitrogen is 3.3%. So nitrogen is present in amino acids. It is present in nitrogenous bases. Okay. Amino acid, they combine to form proteins. Nitrogenous bases, they are the part of nucleotide which forms the DNA. Okay. Then this is sulfur. Sulfur is 0.03. In human body, it is 0.3. So some of the amino acid, they consist of the sulfur also. Now sodium, it is 2.8 in earth crust. In human body is 0.2. Calcium, it is 3.6. Human body is 1.5. Magnesium 2.4. In human body is 0.1. And silicon, you can see the earth crust, it consists basically of the silicon, 27.7%. But in human body, silicon is negligible. Okay. So you can see the elements are same, but the percentage or the composition is different. Clear? So you should remember this table, the elements present in the non-living and the living matter. Okay, now let's move on. Let's talk about the analysis of the organic elements. Okay, how can you analyze the organic elements? So there is a simple procedure that you can follow. So what you can do, you can take a living tissue sample. So you can take any vegetable. Okay. So they have taken liver also. They have given liver as an example also. This is also an organic tissue, you can say. Okay. Then you can grind these tissues in trichloroacetic acid using a mortar and a pestle. So after grinding, there will be a thick slurry will be formed. Okay, so this is, you can see, this is the grinding of the tissues being done in mortar and pestle. Okay, like you grind spices at the home, for the living tissues in the labs we use pestle and the mortar. So here a thick slurry will be formed. Okay, now you can strain this slurry through the cheesecloth or cotton. Okay, now you will obtain two fractions. So when you pass this slurry to cheesecloth or cotton, you will get two fractions. First is the filtrate, which is acid soluble pool. So you have added acid in the beginning. Okay. So filtrate which passes through the cheesecloth, that is the filtrate. Okay. It is the acid soluble pool. Then it contains thousands of organic compounds, some inorganic compounds like sulfate and phosphate as well. Then retentate is the material that don't pass through the cheesecloth, that don't pass through the filters, you can say. So retentate is the acid insoluble fractions. So there are two fractions, filtrate and the retentate. You should remember, filtrate is the acid soluble, retentate is the acid insoluble fraction. Now the retentate contains only 4 types of organic compound, that is proteins, nucleic acid, polysaccharides and lipids. So basically these are your biomolecules. Proteins, nucleic acid, which consists of both DNA and RNA, polysaccharides and the lipids. Now you can extract the compounds from the sample now. You can extract the biomolecules from the sample. You can use various separation techniques to isolate the single compound. So there is one technique known as column chromatography. Okay, you will read about that in higher classes. So you can use that technique, and you can use different solvents also. You can use chloroform, okay, or you can use a mixture of chloroform or some other organic solvent. Okay, so you can use these solvents to separate the biomolecules. You can separate even a single compound. Okay, then you can purify the compound by removing all the other compounds. Okay, so now you have got the single compound now. Okay, now you can use analytical method to determine the molecular formula of this compound. So now you have single compound with yourself. Okay, you have the purified compound. So now using analytical method, so there are a lot of instruments, you can say there are a lot of softwares, okay, through which you can determine the molecular formula of that purified compound. You can even detect the structure of the compound. So a lot of research is being going on where you isolate a single compound from an extract. Okay, suppose you are analyzing a plant extract or fungal extract. So from this extract, using the separation techniques, you can isolate a single compound, and then you can determine the molecular formula of the compound and the structure of the compound. So there is one technique known as NMR. Using NMR, you can detect the structure of the compound. Now, all carbon compounds obtained from living tissue are called biomolecules. Okay. So compounds obtained from the living tissue, they are called biomolecules. Now, how can you analyze the inorganic elements? Okay, first is the ash analysis. So a small amount of living tissue is weighed. Okay, so this is known as the wet weight. Wet weight means along with the moisture. Okay, so this living tissue will consist of the moisture. So that's why we are calling it as wet weight. Then it is then dry to evaporate the water. Okay, so you dry this living tissue now. Okay, so you can use BODs to dry these tissues. Okay, now the water will evaporate, the moisture will evaporate, and now you can again weigh this living tissue, and now you will get the dry weight. Okay, so dry weight is the actual weight. Okay, it doesn't consist of the moisture, so dry weight is actually the accurate weight. It is free of the moisture. Now, if you subtract the wet weight by the dry weight, it will give you the weight of the water content in the tissue. So if you want to determine the weight of the water content, you can simply subtract the wet weight with the dry weight. Now the remaining dry material is fully burned to oxidize all the carbon compound. Okay, so now you have the dry living tissue. So this dry living tissue can be fully burned to oxidize all the carbon compounds. Okay, so when you burn this, then you get the ash. So the remaining powder is known as ash. Okay, now this ash will contain all the inorganic elements like calcium, magnesium, and the other inorganic elements as well. Okay, so therefore elemental analysis gives elemental composition of living tissue in the form of, so if you analyze this ash, okay, using certain instrument, then you will get the following elements in the living tissues. So we have created this ash using living tissues. So the elements can be hydrogen, oxygen, chlorine, okay, carbon, and many more. So we have earlier seen in that table the elements present in the living organisms. Okay, the first method was ash analysis. Now, how to analyze chemical composition? We are studying this. Now, analysis of the compounds gives an idea of the kind of organic and inorganic constituents present in the living tissues. So here is a list of representative inorganic constituent of the living tissues. Okay, so these are the inorganic constituents. Okay, sodium, potassium, calcium, magnesium, water, and some other compounds. These are the inorganic constituent. Okay, so you have to remember all of these table: organic constituents, inorganic constituents present in the living tissues. Okay, so sodium, the formula is also given Na plus, potassium is K plus, calcium is calcium 2 plus, magnesium 2 plus, water is H2O, and then you have these following compounds. Clear? Now let's talk about the structure and properties of the amino acids. Now, organic compounds with an amino group and an acidic group as substituents on the alpha carbon. Okay, we are talking about the amino acid. Amino acid, they are the building blocks of the proteins. Amino acid, they join together through which bonds? They join together with the help of peptide bonds to form a complete polypeptide, you can say, which is known as the protein. Okay, so basically amino acids, they are the organic compound with an amino group. Okay, so they have an amino group, NH2 group, and an acidic group. Okay, as substituents on the alpha carbon. Clear? So they have an NH2 group and they have a carboxylic group. Clear? So amino acid has both of these groups. So they are also known as alpha amino acids because these amino group and acidic group, carboxylic group, they are substituents on the alpha carbon. Now, this is very interesting. These amino acids, they are also called substituted methanes. Okay, why they are called substituted methanes? Okay, so what is the formula of methane? Methane is CH4. Means there is a single carbon atom. Okay, and there are 4 hydrogen atoms. So single carbon atom is joined to, you can say, or bonded to or attached to 4 hydrogen atoms. Okay. So why it is called substituted methane? Because the 4 hydrogen atoms, they are substituted with other groups. Okay. What are these other groups? Okay. One is hydrogen. Okay. Then second is the carboxylic group, which is COOH. Then the amino group, which is NH2. And there is an R group. Okay. R group can be variable. R group, you can also call R group as the side chain. So R group can be your methyl, okay, ethyl. Okay. That's why it is written variable R group. Okay. So now you can see why it is called substituted methane. So in the center, there will be carbon. Okay. And then you have hydrogen on one side. You have carboxyl on other side. Then you have amino group, and then you have the R group. Now let's talk about the type of the amino acids. Now based on the R group, there are many amino acids. Okay, only 20 types occur in the proteins. So basically there are 20 types of amino acids. Okay, these proteins, they are made up of 20 types of amino acids. Okay, so you must have studied that there are 64 codons. Okay, although we have 64 codons, but the amino acids are only 20. So this means that more than one codon is coding for the single amino acids. Okay. Now what can be the examples of the variable R group? So you can see if the R group is hydrogen, okay, then it is the glycine. So glycine is the simplest amino acid. Then if the R group is the methyl group, then it is the alanine. Okay. If the R group is hydroxymethyl, then it is serine. Then you can see these are the figure image showing you amino acids. Okay. Here, if the R group is H, then it is glycine. If the R group is CH3, which is the methyl group, then it becomes alanine. Then in serine, you have hydroxymethyl. Hydroxy means OH group. Okay, CH2OH is hydroxymethyl. So then it becomes serine. Okay, so you can see this is a substituted methane. Methane have C and then 4H. So this amino acid, this had one H group, one carboxylic group, amino group, and this is the variable R group, which can also be called as side chain. Now let's talk about the classification of the amino acid based on the functional group. Okay, the first one is acidic amino acids. Okay, for example, glutamic acid. So what are these acidic amino acids? So these amino acids have a negatively charged side chain. Okay, that's why they are called acidic amino acids. Example is glutamic acid. Another example is aspartic acid, asparagine. Okay, these are all acidic amino acids. Now what are basic amino acids? So these amino acids have a positively charged side chain. Okay, so they have a positively charged side chain. The second one is basic amino acids. Example is lysine, arginine. These are all basic amino acids. Then you have neutral amino acid. Example is valine. So these amino acids have side chains that are neither acidic nor basic at the physiological pH. Okay, so these are neutral amino acids. They have neither acidic nor basic charge. Then you have aromatic amino acid, which are in the form of rings. Example is tyrosine, phenylalanine, and tryptophan. So here you can see these are the types of the amino acids. Okay, clear? So you have arginine, histidine, lysine, aspartic acid, glutamic acid, serine, threonine, asparagine, glutamine, glycine, proline, cysteine, and selenocysteine. You have alanine, valine, isoleucine, leucine, methionine. Okay, you have phenylalanine, tyrosine, and tryptophan. Okay, and you can see the abbreviation of this amino acid is also given to you. Clear? Like for histidine, you can write HIS or simply H. Okay, so there are 20 types of amino acid. Some of the amino acid also consist of sulfur as well. So there are sulfur containing amino acids also. Okay, now let's talk about the ionizable nature of these amino acids. So see, this amino acid have two groups, NH2 group and the COOH group. So these groups can ionize. Okay, so because of this ionization, there are structural changes in the solution of different pH. Okay, so when you add these molecules at different pH, then there are some structural changes. Okay, so deuterion is formed. So what is deuterion? Okay, so an amino acid can exist in an ionized form. Okay, and in this ionized form, both positive and negative charge occurs. Okay, now this form is known as a deuterion. Clear? It has a both positive and a negatively charged, but a net zero charge. Okay, net charge is zero. So you can see here this B, this is the deuterion here, this one. It has a positive charge also and it has a negative charge also, but the net charge is zero. Okay, so this structure B, this is the deuteronic form. Now let's talk about another biomolecule, which is lipids. So lipids, they are generally water insoluble. Okay, they could be simply fatty acids. So lipids can be simply called as fatty acids. So what is fatty acid? Okay, the fatty acids are basically, what is the structure of fatty acid? They consist of a carboxyl group which is attached to an R group. This is fatty acid. Okay, carboxylic group will be attached to an R group. Now R group can be variable. Okay, so R group can be, it can be a methyl group. Methyl group is attached to the carboxyl group. Then ethyl group, this is attached to the carboxyl group. Now, okay, this you can see the number of carbon can be from 1 to 19 carbons. So here you can see these are lipids. Lipids are also, they can be called as fats and oil. We'll talk about the difference between fats and oil also. Okay, so these fats and oil, these are both lipids. So you can see this is the fatty acid here, like palmitic acid. So fatty acids are nothing but the R group is attached to the carboxyl group. Then you can see glycerol. Glycerol is a kind of alcohol. When glycerol combines with fatty acid, then you get the glyceride. Okay, so triglyceride consists of 3 fatty acid and 1 glycerol. Then you have diglycerides, you have monoglyceride. Okay, then you have phospholipids like lecithin. Lecithin example of phospholipid. They are basically present in the plasma membranes. Okay, they consist of this phosphate group. You can see this is a phosphate group. Then you have this cholesterol. Okay, we were talking about the fatty acids. For example, a fatty acid is palmitic acid. It is made up of 16 carbons, including the carboxyl group. Then arachidonic acid, it consists of 20 carbon atoms, including the carboxyl carbon. Okay, now what are the types? Okay, so they can be saturated fatty acid or unsaturated fatty acid. Saturated means without the double bond. It doesn't consist of a double bond. Unsaturated means with one or more carbon-carbon double bonds. This is the difference between saturated and unsaturated. Okay, this image we have already discussed. We have fatty acids, we have glycerol, we have triglycerides, phospholipids, and this cholesterol. Okay, now let's talk about glycerol. Okay, so glycerol actually It is trihydroxypropane. Okay, what is glycerol? It is trihydroxypropane, means it consists of 3 carbons and 3 hydroxyl group. Okay, that's why 3-hydroxypropane. Now, when lipids combine with when glycerol combines with fatty acid, then glycerides are formed. Okay, so fatty acid, it esterifies with the glycerol, means there is a ester bond formation between fatty acid and the glycerol. Okay, so what happens? The fatty acids combine with the OH of the glycerol to form the ester bond. Okay, this is known as esterification. So that's why it is written fatty acid esterifies with the glycerol. Okay, now what is created when fatty acid is esterified with the glycerol? Then glycerides are formed. Okay, so monoglycerides can be formed diglycerides or triglycerides. So monoglyceride consists of 1 fatty acid and 1 glycerol. Diglyceride consists of 2 fatty acid and 1 glycerol. Triglyceride consists of 3 fatty acid and 1 glycerol. Okay. Okay, these lipids, they are also called fats and oils based on their melting point. Now, what is the difference between these fats and oils? So, oils have lower melting point. For example, gingerly oil, and remain as oil in the winters. Now, fats have higher melting point. Okay, this is an image showing you fats and the oils, which are both lipids. Okay, now let's talk about the phospholipids. So, obviously, the name phospholipids suggests that phosphorus is present. So, they contain phosphorus and a phosphorylated organic compound. Okay, they are found in the plasma membrane, or you can say the cell membrane. They are made up of phospholipid bilayers. Okay, example is lecithin. Lecithin is example of phospholipid. Okay, then they are complex lipids. So these complex lipids, they are found in the neural tissues. So what do you mean by neural tissues? Means the tissue of the brain and the nerves. These are the neural tissues. So basically, these lipids, complex lipids, they are combined with the proteins and the carbohydrates. These are complex lipids. These are present in the neural tissues. So they have more complex structures. Okay. Now next is heterocyclic rings. Okay. So these rings are found in various carbon compounds. Okay. Where are these heterocyclic rings present? So they are present in the nitrogenous bases. You must be knowing about the nitrogenous bases. Okay. So these nitrogenous bases, they are present in the DNA. And DNA is made up of nucleotide. And what is nucleotide made up of? So nucleotide is made up of nitrogenous base, a sugar, and a phosphate. Okay. So nucleotide consists of these 3 components. And what does nucleoside contains? So nucleoside consists of only nitrogenous base and the sugar. It lacks a phosphorus. It lacks a phosphate group. So nitrogenous bases can be adenine, guanine, okay, cytosine, and then uracil. Uracil is present in RNA. Okay, then you have thymine. Okay, these are all nitrogenous bases. They contain nitrogen in them. Okay, so these nitrogenous bases, they consist of heterocyclic rings. Okay, this is adenine, this is purine. So nitrogenous bases, there are 2 types, purines and pyrimidines. Okay, purine pairs with pyrimidine in the DNA. Okay, this is structure of adenine. You can see this the heterocyclic structure you can see. Then uracil, this is a pyrimidine. Okay, it consists of a single ring. Then these are adenosine and uridine. Okay, then you can see this is adenylic acid. Clear? So you have nucleotides and you have nucleosides. These are nucleosides. Adenosine, uridine. Okay, clear? So you understand the difference between nucleotide and a nucleoside? This is very important. Nucleotide consists of a nitrogenous base, sugar molecule, which is the pentose sugar, and a phosphate. Nucleoside, it only consists of sugar and the you can say sugar and the nitrogenous base. It lacks the phosphate group. Okay, let's talk about the nucleoside. So it is formed when nitrogenous base attaches to the sugar molecule. Then examples: adenosine, guanosine, thymidine, okay, uridine, cytidine. Okay, clear? So you should remember the examples also of the nucleosides. Then you have nucleotides. So they are formed when a phosphate group is esterified to the sugar in a nucleoside. Examples: adenylic acid, thymidilic acid, guanylic acid, uridylic acid, cytidilic acid. Okay, these are all nucleotides. Clear? Here phosphate group is esterified to the sugar in a nucleoside, and it also consists of a nitrogenous base. So DNA is a polynucleotide. The nucleic acid, they consist of DNA and the RNA. Clear? So it consists of nucleotides only. Okay, nucleic acid consists of nucleotides only. DNA, you know, it is the genetic material. Earlier RNA was the genetic material. So there was some modifications in the RNA. Okay, and then we got the DNA. So like RNA has uracil. This uracil methylated and it became thymine. So thymine is basically methylated uracil. So you can see this is RNA, this is DNA. So DNA consists of adenine, guanine, cytosine, and thymine, and RNA consists of adenine, guanine, uracil. Okay, instead of thymine, you have uracil in the RNA. Okay, it has guanine, cytosine, adenine as well. You can see DNA is double stranded. That's why DNA is more stable. RNA is single stranded. Although RNA can also become double stranded, but usually RNA is single stranded only. Even messenger RNA, that is also single stranded. Okay, DNA is double stranded because of the complementarity, base pair complementarity. Okay, one strand is in 5' to 3' polarity, other strand is of the reverse polarity. So both of these strands, they are anti-parallel to each other. Okay, now let's talk about the primary and the secondary metabolites. Okay, so these primary metabolites, they are found in all living organisms, including animals, plants, fungi, and the microorganisms. These primary metabolites, they help in the growth, they help in the development of the organisms. So these include amino acids, sugars, and other essential organic compounds. Okay, amino acid, they form the proteins. Okay, now they have identified functions in the normal physiological processes. So basically they have some functions. They help in the growth and development of the organisms. Then you have the secondary metabolites. So predominantly found in plants, fungi, and the microbial cells. So secondary metabolites, they are basically the byproducts of the metabolism. Okay, examples of metabolites are alkaloids, flavonoids, rubber, essential oils, antibiotics. These are also secondary metabolites. Then certain pigments, scents, gums, and spices. These are all secondary metabolites. Okay, these are the byproduct of metabolism. So you can see these antibiotics. or these secondary metabolites, they are produced by these organisms, plant, fungi, and microbes. Okay, so these are produced as a defense mechanism. Okay, they are not being produced for human benefit, so they can benefit humans. They are not producing it for human welfare. They are producing it as a defense mechanism. Okay, now exact biological role in the host organisms are not fully understood. Okay, so mostly they are being synthesized as a defense mechanism, but exact biological roles of most of these secondary metabolites is not fully understood. Now, many are beneficial for human welfare also, like rubber, certain drugs, which can be used as medicines and antibiotics. Like you must have seen some of the antibiotics have been isolated from fungus and from the bacteria, like streptomycin has been isolated from streptomyces, this bacteria. Okay. Then penicillin has been isolated from penicillium. Then spices, scents, pigments, these can be used by the humans. Okay, some have ecological significance also. What do you mean by ecological significance? See, certain microorganisms, certain fungi and bacteria, they can help in mineral cycling, or you can say nutrient cycling. Okay, so they are decomposers. They decompose the organic matter and they help in the nutrient cycling as well. So they have digestive enzymes. They secrete the digestive enzymes and they can degrade the organic matter. Now, this is a table showing you some secondary metabolites. So you can see here the pigments, alkaloid, terpenoid, essential oil, toxin, lectins, drugs, polymeric substances. These are all the secondary metabolites. And then examples of the pigments: carotenoids, anthocyanins, alkaloids are morphine and codeine, terpenes are monoterpenes, diterpenes, essential oil, lemongrass oil, and toxins are heparin and ricin, lectins are concanavalin A, and drugs, vinblastine and curcumin, and polymeric substances are rubber gums and cellulose. Okay, now let's summarize this entire lecture. We have discussed the theory. Now let's discuss the summary. How you can analyze the chemical composition. So basically, chemical analysis of living tissue reveals elements such as carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. These are the major components present in the living tissues. Remember this. Now, some other inorganic elements present are sodium, potassium, calcium, and magnesium. Okay, carbon is the basic element in all the biomolecules. Biomolecules are your DNA, proteins, carbohydrate, lipids. So carbon is the basic element here. Then you have oxygen and hydrogen also. Okay. Biomolecules are organic compounds present in the living organism, for example, protein, carbohydrates, and lipids. Now, most of the biomolecules are organic compounds containing carbon, hydrogen, oxygen, and nitrogen as the primary elements. Okay. Some biomolecules also contain phosphorus and sulfur in addition to the primary elements. Okay, like DNA consists of phosphorus. Okay, and then this amino acid consists of this sulfur. We talked about the primary and the secondary metabolites also. The biomolecules can be broadly classified as primary and secondary metabolites. The primary metabolites, they are directly involved in growth and development. Okay, examples are protein, nucleic acid, carbohydrates, and lipids. Then secondary metabolites, they are the byproduct of metabolism. They are not directly involved in growth but play ecological roles. Examples are alkaloid, terpenes, resins, and tannins. Now let's discuss some NCERT based questions. Okay, what is the first question? What is the most abundant element by weight in a living organism? Okay, what are the options? Carbon, nitrogen, oxygen, and hydrogen. What is the correct answer? So let's have a look at the answer. The answer is option number C, oxygen. The composition by weight in living organisms: oxygen is approximately 65 to 67%. Carbon is approximately 18 to 90%. Then you have hydrogen, which is approximately 10%, and nitrogen is approximately 3%. Okay, option number C is the correct answer. Next, which of the following is a characteristic of biomolecules in the living tissues? Okay, let's have a look at the options. Option number A, they are always small molecules. Okay. Option number B, they are exclusively inorganic. Option number C, they are primarily composed of carbon, hydrogen, oxygen, and nitrogen. And option number D, they are not involved in any chemical reactions. What will be the correct answer? So let's have a look at the answer. C, option number C is the correct answer here. They are primarily composed of carbon, hydrogen, oxygen, and nitrogen. Now, carbon forms the backbone of the organic molecules. Hydrogen participates in various bonds and contributes to molecular stability. Like in DNA, you can see the nitrogenous bases. They are joined together via hydrogen bonds. So between A and T, there are 2 hydrogen bonds. Between G and C, there are 3 hydrogen bonds. Okay. Now, oxygen is crucial for many functional group and cellular respiration as well. And nitrogen is essential for proteins and the nucleic acid. Okay, the proteins are made of amino acid. Amino acid consists of an amino group, and nucleic acid also consists of the nitrogenous bases. Now, many biomolecules are large, means they are macromolecules. Okay, examples are protein, DNA, and some complex carbohydrate. These are macromolecules. Most biomolecules are organic compounds. And biomolecules are constantly involved in chemical reactions essential for the metabolism. Now, next question. Which of the following is a primary metabolite? Okay, these primary metabolites, they are involved in growth and development. Options are alkaloid, rubber, amino acids, and morphine. What is the correct answer? देखिए option number C is the correct answer, amino acids. Okay, this is the primary metabolite. See, primary metabolite, they are directly involved in normal growth, development, and reproduction of the organisms as well. Okay, amino acids, they are essential building blocks of the proteins. Okay, they are required for basic cellular functions as well. Okay, so these proteins, okay, lot of enzymes, they are made up of proteins. The proteins, they carry out a lot of functions. They act as a catalyst also. They can speed up the reaction also. They convert substrate into products. Okay, they are involved in a lot of biochemical reactions. They are involved in a lot of metabolic pathways. Now, alkaloid, rubber, and morphine, these are the secondary metabolites. Clear? They are produced as a byproduct of metabolism as well. Okay, so plant or the organism, okay, or the microorganism, they are producing this as a kind of defense mechanism also. Okay, now let's discuss some keynotes. Okay, now the most abundant chemical in living organisms is water, which is H2O. The 4 major classes of biomolecules are carbohydrate, lipid, proteins, and the nucleic acids. Okay, these are the 4 category of biomolecules. Then amino acid exists as deuterines in the solution. Deuterine consists of both the charges, positive and a negative, but the net charge is 0. Okay, so this you can see when you add this amino acid in solutions of different pH, then the structure can change and a deuter ion can form. Clear? So we'll talk about these biomolecules also. Okay, we'll talk about nucleic acid also in our upcoming lectures as well. Now there are some interesting questions for you. First question is, why are biomolecules considered the building blocks of the life? Okay, obviously they are directly involved in growth and development. Then next question, why it is essential to analyze the chemical composition of the living tissues. The next one, why are biomolecules referred to as the organic compounds? And then fourth one is, how does the primary structure of a protein influence its final 3D structure? Okay, so proteins have a primary, secondary, tertiary structure, even quaternary structure. Okay, so primary structure is the linear sequence of the amino acids. So how this primary structure is influencing the 3D structure? So in the 3D structure, the protein, the enzyme, they are active. Okay, the next one, why are primary metabolites considered essential for growth and development, while secondary metabolites are not directly involved in growth and development? So you have to justify this statement, you have to comment on this statement. Okay, so this was the end of this session. Okay, we talked about the analysis of the elemental composition of the living tissues. Okay, we talked about the you can say amino acids. Here we talk about the lipids as well. Okay, so these are all biomolecules. So with this, we end this session. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.